Okay, good morning or afternoon, everyone. Sorry for the delay, a little bit of technical difficulties. So welcome to the second session of the 2019 Cliff Grads webinar series, which we've titled Direct Measurement Approaches to Investigating Mitigation Options in Livestock Systems. So as I mentioned during our first webinar, this series is designed to allow Cliff Grads Fellows the chance to share your research, learn more about the science of low emissions development and research approaches to investigating mitigation options, build research and career skills, and also start to get to know each other a little bit. Today's webinar features 3 Cliff Grads Fellows who undertook fellowships in 2018 and who are all investigating greenhouse gas emissions and mitigation options in livestock systems. Unfortunately, one of our presenters couldn't join us, but our first presenter today, Florencia Garcia, will discuss her work on approaches to mitigating enteric methane emissions from cattle, specifically using approaches to engineering the rumen of the cattle. Our second 2 presenters, Maria di Bernardi and Banira Lombardi, will present results from their work on greenhouse gas emissions from pastures. Finally, at the end of the webinar, I will share some tips for writing and getting your research published in academic journals. I know that some of our panelists and participants also have experience publishing in academic journals, so if you have a question or a lesson especially that you've learned along the way, or a favorite tip for academic writing and publishing, please share it in the question box, and I will share them all during the discussion about writing and publishing at the end of the presentation. And you can share those questions at any time. Next slide. A little more about our presenters. Florencia Garcia is a PhD student in ruminant nutrition and an assistant professor at the National University of Córdoba in Argentina. During her Cliff Grads Fellowship, she was hosted at the Institute for Agricultural Research, INEA, in Osorno, Chile. She will be discussing the effect of inhibiting methane production in the rumen on nitrogen use efficiency. Our second presenter, Maria de Bernardi, is completing her doctoral degree in applied sciences, specializing in environment and health at the National University of the Center of Buenos Aires in Argentina. She spent 6 months at the Institute for Agricultural Research, INEA, also in Osorno, Chile, and will be presenting her work on evaluation of greenhouse gases in pastures with foliar N fertilizers. Lastly, Banira Lombardi is a PhD student in applied sciences at National University of the Center of Buenos Aires, also in Argentina, with Maria, and she is specializing in greenhouse gas emissions from livestock effluents. Her presentation is titled, Can Pasture Type Influence Nitrous Oxide Emissions from Cattle Manure? Finally, my name is Meryl Richards, and I am the Science Officer with the Low Emissions Development Flagship of the CGIAR Research Program on Climate Change, Agriculture, and Food Security. I have a PhD in Plant and Soil Sciences from the University of Vermont, and I am still based here at the University of Vermont in the United States. So now I'd like to hand the floor to Florencia. Florencia, go ahead.
Hi. Can you see my presentation?
Yes, perfectly.
Great. Hi everybody, my name is Florencia Garcia. I'm from Córdoba, Argentina. I visit the Agricultural Research Institute in South Chile, and the research I participated in to try to answer the question, what is the effect of inhibiting methane production in the rumen or nitrogen use efficiency? Well, ruminants have the advantage of take forage fiber as a feed because they have a consortium of microorganisms that lives in the rumen. The rumen is an anaerobic environment, and in these conditions, the reduced cofactors need to be reoxidized in order for fermentation to go on. There's different mechanisms for this to happen, but in the rumen, the main electron sink for this process is methane. Methane is a potent greenhouse gas with 28 times more global warming potential than CO2, as well as it implies for the animal a loss of the energy that it obtains from the feed. So there's important ongoing efforts to reduce methane production. But when we reduce methane production, there's an accumulation of hydrogen and an increase of the rate of reduced to oxidized cofactors. That actually means that there's a change in the rumen environment. So the question that may arise is what happens with other metabolic pathways when the hydrogen dynamic has changed because of methane has been inhibited? The research I participated into had the aim to understand how inhibiting methane production in the rumen affects microbial protein production and its implication to the efficiency of nitrogen utilization of dairy cows. The hypothesis that lies in this idea is that the inhibition of methane production will result in an increase of the abundance of producing equivalents that will stimulate an increase in microbial protein production in the rumen, improving the nitrogen use efficiency. And where the implication of this work, this research focused on understanding the interface between the energy and the nitrogen metabolism in the rumen as a part of the interaction between the fermentation and the anabolism, which has been kind of little explored, especially in vivo experiments. The research was conducted in the unit of digestion and metabolism in Iña, Osorno, South Chile. We evaluated 4 treatments, 2 related to the methanogenesis inhibition, yes or no, and 2 different protein sources. Cannulated lacting cows in early lactation. The diet was based on grass silage, cornflake, rapeseed meal, and mineral sodium vitamin premix, and urea in those treatments that had the inorganic source. The experimental design, the 8 cows were divided in 2 groups, which received a different diet. The difference was 4 cows received only the rapeseed meal, while the other 4 cows received the inorganic source of protein, as well as rapeseed meal. We have 2 different periods. In the first period, which was the control period, there was no inhibition of methane, and in the second period, we inhibit methane production in the rumen. Both periods had a period of adaptation, both to the diet or to the additive, and 6 days of measurements. In those 6 days, the variables that we evaluated were related to microbial nitrogen production with 3 different methods. Methane was evaluated with using the SF6 technique. Rumen environment, we evaluated BFA, ammonium, pH, the redox potential, microbial long-chain fatty acids, and the composition of the microbial community. We also evaluated animal performance as for meal production and composition, as well as changes in weight and body condition of the cows. And other variables that we evaluated were transmitted intake, nutrient digestibility, metabolizable energy, nitrogen balance, as well as blood metabolites. I don't have any results so far to share with you today, so I think my message for today, the takeaway message will be that methane plays a biological role in the rumen. It is strictly related to the hydrogen dynamic, and the hydrogen dynamic in the rumen is also related to BFA production with the microbial mass and long-chain fatty acids, as for example. And these 3 examples in the end are the sources of energy and protein for the animal. So we don't have to forget that to take knowledge of the implication of inhibited methane production. Where are the processes may be affecting when we inhibit methane production? So going back to our cow in the beginning, we can consider, for example, positive effects if we're changing long fatty acid metabolisms and improving the quality of fat in the meat and milk, for example. Or we may be thinking of negative effects if we're, by inhibiting methane production, enhancing nitrogen release, for example, in urine. So we don't have to forget to do, when evaluating mitigation strategies, to do them as broad as possible, not only target one product. And my advice for future Cliff Guard students will be at least to share what my experience was. And in my case, I had the opportunity to have a lot of contact with my hosts from even before going to my visit. So it was enriching to have a deep knowledge of the hypothesis and the work proposal before my arrival. This allowed me to discuss in depth the conceptual framework of the experiment, the methodology, and to take full advantage of my time, because it allows me to organize myself what to do with the time once there. And I think it is a great opportunity to interact with other researchers, not only those strictly related to the work you are going to participate into, but look up, take the time to meet people, to meet places. You're in a different country, to know other different costumes. So I think, or at least how I live, is every moment is a chance to do something new. So take that opportunity. In the end, thank you, especially to my host, Camila Munoz and Emilio Ungerfeld, who received me with very, very, very good. I'm pretty happy. And also to the program. I'm very grateful for this opportunity. Thank you.
Thank you so much, Florencia. So now we'll hand things over to Maria. And in the meantime, I want to say I really appreciate your comment about your interaction with your host before your visit. I think that's great advice for any students who haven't yet undertaken their fellowships, to really do some pre-planning and make sure that you can make best use of your time there. So thank you for that. Maria, go ahead.
Hi everyone. I'm María Bernardi. I'm from Tandil, Argentina, and I was in Osorno, Chile during my Cliff Guard stay. I participated in the project nitrous oxide emissions from pastures using novel fertilizer formulations. In particular, increased nitrogen use efficiency in productive systems implies an economic and productive efficiency. This nitrogen can be lost to atmosphere as nitrous oxide, as nitrogen monoxide, ammonium, or nitrogen as a gas. So we can limit these loss, improving the NUE. It was proved that the use of foliar and fertilizer as urea dissolving water in pastures foliage increased the NUE compared with normal urea. In spite that nanoparticular fertilizer was already proved its potential, but it's not common the use of foliar and nanofertilizer in pastures. So the major goals of this project are increase pasture NUE using a foliar, nanoparticular, and fertilizer in pastures, and avoid and loss to environment, determining end balance and end cycle. During my stay, we measure nitrous oxide emission and ammonium volatilization, and also I was able to contribute with the measure and interpretation of methane fluxes. For that, 2 essays was carried out with soil scores collected in Osorno, Chile, a pre-essay with soil collected in Niña Remeue, and the principal essay with soil from Iña La Pampa. In both cases, soil scores were put in a controlled temperature room, and humidity control was carried out during whole study period, maintaining 50% of water holding capacity. The pre-essay was carried out principally to learn how to use the different methodologies and to adjust some parameters that we need in the principal essay. For that, the treatment applied were in the pre-essay a control with plants and 4 foliar fertilizers, 2 nano-fertilizers, 1 commercial and the other created in India labs, an ammonium nitrate fertilizer and urea dissolving water. While in the principal essay, a control without plants and granular urea were added. For measure of methane and nitrous oxide fluxes, a variant of static chamber technique was used, and for ammonium volatilization, we use a dynamic chamber technique. And also some parameters from soils and plants were measured to characterize soil, pasture, and to calculate NUI. From pre-essay, we could obtain some considerations to adjust measurement parameters for principal essay. We can see in this figure the methane fluxes along 21 days of measurement. We found that we need 1 hour to obtain the methane flux from each core soil, and we didn't find any difference into treatment respect methane flux. But we can see that all soils had an uptake behavior. For nitrous oxide, that we see in the right figure, then we can see that the major emissions were registered in pastures with ammonium nitrate fertilizer. And in all cases, the emissions were increasing a long time, and for that we decided to extend the study period to 60 days. Also, waiting period was fixed every 2-3 days. And with all of that, in the principal essay, I have no result yet, but we expect that the measured emissions of nitrous oxide come from granular urea and ammonium nitrate, and the lower emissions were expected for end-foliar nanoformulation. This also is expected for ammonium volatilization. And this result could help to take more careful decisions about which fertilizer is convenient to use in pasture systems and help to develop a new and better fertilizer. Finally, I advise you to get involved completely in fieldwork as an organizer and in person in charge. This gets you experience in organizing field or lab research and get in charge of it. This includes the analysis and discussion of results that make you understand better our work and the decision that we can suggest in order to reach a more eco-conscious agriculture. And for that, you need to continue in touch with your host researchers and your partners. Well, that's all. Thank you.
Thank you so much, Maria. Now I'd like to hand the floor over to Banira for her presentation.
Hi, my name is Banira from Tandil, Argentina, and I spent 4 months in Colombia at the International Center for Tropical Agriculture, working to answer the question, can pasture type influence nitrous oxide emissions from cattle manure? The purpose of my research was to understand the interaction between tropical grasses and cattle excreta. Animal production systems are important sources of greenhouse gases. When grazing cattle defecates, urine and manure are deposited on pastures, leading to the formation of methane and nitrous oxide. Some tropical pastures have the capacity of reducing nitrous oxide emissions through the exudates that release their roots. These compounds inhibit soil nitrifier activity, which is known as biological nitrification (BNI) process. A previous study demonstrated a reduction in nitrous oxide emission from cattle urine deposited on high BNI tropical pastures. So, the aim of my research stay was a follow-up to the urine study to see if there is a BNI influence on gas emissions from cattle manure. Our hypothesis was that tropical pastures with high BNI capacity can reduce nitrous oxide emission from deposited manure. For the experimental site, we used a part of a long-term experiment established in 2004 to evaluate the BNI potential of several tropical pastures. Here we select two contrasting grazes. The first of one with low BNI capacity, which is Bracharia mulato, and the other one was Bracharia thuli with high BNI capacities. On each parcel, we deposited fresh manure simulating a natural deposition, and to evaluate gas emission, we used the static chamber technique during 51 days. There were 2 chambers with manure and 1 free of manure as a control, being a total of 18 chambers. The experiment was done in a dry season, and methane, nitrous oxide, and carbon dioxide emissions were quantified. Gas monitoring was complemented by the measurement of temperature and humidity. Also, manure and soil were sampled to study some parameters like the total carbon and nitrogen concentration, the mineral nitrogen cycle for ammonium and nitrate determinations, the potential nitrification rate to verify the BNI effect, the microbial abundance of nitrophagous archaeas and bacteria, and macrofauna. As we can see in the graph, related with the mineral nitrogen cycle, at the beginning ammonium was greater on manure than soil, and then it decreases. On the other hand, nitrates on manure increased, indicating that nitrification is happening, and then go to background values. The behavior of all the parameters were similar in both treatments, and nitrification rates, microbial activity, and macrofauna are still in analysis. Manure emissions were greater in comparison with control, but carbon dioxide, methane, and nitrous oxide emissions were similar among treatments. Although we expected to find differences on nitrous oxide emissions, like in the urine study, it didn't happen. We supposed that there was not influence because the manure dried fast and the nutrients never reached the roots where the BNI effect exists. So we think, what would happen in wet conditions? To test it, we developed a second essay of 35 days, only to gas measurement, depositing new fresh manure on pastures and adding water to simulate wet conditions. The second experiment resulted in lower nitrous oxide emissions from manure on thuli compared to mulato, indicating a reduction trend according to our hypothesis. In any case, resulted a nitrous oxide emission factor lower than the IPCC default value. So we conclude that tropical pastures with BNI capacity represents a low emission technology that can play a key role in mitigating nitrous oxide emissions from cattle manure, especially under wind conditions. These are preliminary results, so more studies on cattle manure emissions will be needed, evaluating the effect of quantity and quality of manure and environmental conditions. Finally, my research stage was an incredible experience. It will make you grow personally and academically, testing your previous knowledge and also your personal skills of adaptation, tolerance, and open mind. So you have to be prepared. Enjoy it because I think it is one of the best experiences that can be done in a PhD training. Now I'm back home with my backpack full of learning experiences, memories, and friends. Thank you.
Thank you so much, Banira. So now we'll have a chance for all of our participants to ask questions of our panelists. And the way that you can do that is the same as during the last webinar. Just enter your questions in the questions box in the GoToWebinar control panel. And I will read them out and give our panelists a chance to respond to them. I also want to request that if you have any suggestions in terms of writing and academic publishing, you can also enter those at any time during the webinar, and we'll discuss those after the final presentation. So to start with, I have a few questions for our presenters. First of all, Florencia, I'm curious if you could explain the SF6 technique a little bit more, because I know that some of our CliffGrads 2019 participants may also be using this technique, but some of them might not be familiar with it. And can you also discuss any challenges that you have run into using the SF6 technique and how you've addressed those? So Florencia?
Yep. Thank you for the question. In the past, I've been involved in Argentina. There's not many groups that we have the SF6 already functioning, so it was an invaluable opportunity to put it in practice. The SF6 has the principle. The SF6 is sorry, cannot say good in English, but hexafluoride of hydrogen is a tracer. That tracer goes in the rumen with a permeation tube. That permeation tube have the SF6 inside. Once in the rumen, with a temperature in the rumen, it releases at a known rate. That known rate is determined in advance before putting the permeation tube inside the rumen. We put the tube into an oven for, say, 8 weeks, and twice a week we measure the change in weight. That way we can determine the permeation rate, and once we know the permeation rate, we know how much SF6 is releasing by time. So we have the SF6 releasing. We capture the SF6 with a tubing that is in the nostrils of the animal. We have a canister that is in vacuum. That canister in vacuum is taking what the animal is breathing out. A part of the animal is breathing out, and that then we analyze it by gas chromatography. We determine the SF6 as well as the methane. Knowing the quantity of SF6 and the rate, the permeation rate, we can determine the volume of gas that is being released by day. And knowing the volume and the concentration of methane that is in that gas, we can determine the gas production, the methane production, sorry, in a day. That's the principle of the SF6 techniques. The SF6 techniques have many small details that all of them are very important, and that's the main challenge, because the SF6 can have it's a great technique when used with care, I will say. In the group that I participate into, Camila Munoz has long experience in this. Her first year in using this technique were difficult, but nowadays she has the technique. She knows how to use it. But I think the main problem that I have, or my challenge, was you have to be able to regulate the rate that the gases that go into the canister, and to do that was one of the problems. We have sorry, no, I don't remember the word, but to reduce the amount of gas that goes into the canister. That was one of the complications that I had to determine that part. But I think that the main challenge is to try to do it a first time, and the first experiment with the SF6 can be quite tricky. I hope to have answered the question.
Yes, that's great. Thank you so much. So we have a couple more questions coming in. One from Yuri, and the question is for Banira. And he asked, Banira, do you plan to test different cow species?
Sorry, I don't understand. Can you repeat it?
He asked if you plan to test different species of cows. So manure from different species of cows.
No, it's just the same species.
Okay, so just one species that you're working with?
Yeah.
Okay, thank you. We have another question from Rita, and his question is to Florencia again, which is, what were the microflora investigated? And what happens for the microflora studied during the measurement period?
Thank you again for the question. The main groups that will be targeted are protozoa and methanogenic archaea, and some of the main groups of bacteria. But this still is under analysis, and we still have yet determined exactly. That's a cost-related issue. What exactly groups of bacteria we're about to determine, but in the principle, there will be main archaea bacteria, protozoas, and one of the main groups of fibrolytic bacteria. Could you repeat the second question, please?
Sure. The second part of the question was, just finding it here. What happens for the microflora that were studied during the measurement period?
We have rumen cannulated cows, and that can be a difficulty. Consider that you have oxygen going into the rumen, so that's a limitation of this approach, but that's a risk that we were ready to have. Even though we know that had limitation, that is a risk, yeah. But there's more information you can take out of having a straight link to the rumen and having samples from the rumen, that they are disadvantaged that you have that oxygen is going in.
Okay, thank you. I have one question for Maria, which is, are foliar and nanofertilizers commonly used in the study area? And how do they compare in terms of cost with ammonium nitrate? I realize that's a little bit outside of your study topic, but I'm curious if you have any information about that.
Yes, we use these fertilizers that I mentioned. The common fertilizers are urea, just urea in the field, but urea dissolving water is getting common. And the other common fertilizer is ammonium nitrate, are the commercial ones that are common in also in Argentina, in Chile and in Argentina. Compared with nanofertilizers, the nanofertilizers are new in this field, so maybe I don't know the cost really. This other part of the group in Chile were studied that, but if these fertilizers increase in the new efficiency and increase the production that you have, that can maybe get that the cost is not so high. So you can understand me, maybe?
Yes. Yeah,
I don't know if I respond the question, but
yeah, that makes sense. So you potentially expect them to increase productivity.
yeah.
Okay, great, thank you. Another question from Titus Abdini. Two questions for two different speakers. So first to Florencia. Can you explain the mechanism or pathway when methane formation is inhibited and leads to more nitrogen emissions in urine? So that's a question for Florencia.
Thank you for the question. That would be an example if, for example, this is not a regular method to, or there's a mitigation strategy, not in urine. That is not a mitigation strategy, but I think the message was that you have to take care of other processes in the rumen. And that was to set an example that if you're targeting to inhibit methane production, but in the same way you're increasing another contaminant, that will be unfavorable. Using nitrate as reducing agent is not a mitigation strategy, it's a proof of concept. It just was the idea to not lose in mind that when reducing one element, we cannot increase another one.
Great, thank you. And let's see, second question, also from Titus to Banira. Do you expect that different types of tropical grasses will release different emissions due to nitrous oxide application? And do you suggest to revise the emission factors for nitrous oxide that has been developed by the IPCC according to the type of forage? So that's to Banira.
Can you hear me?
Yes.
Okay. I don't know if I understand right the question, but with the total nitrogen applied of the dung patch, the manure patch, at the end I could calculate the emission factor, and all the results were lower than the IPCC. And I don't know, the first question, can you repeat me? Sorry.
Sure, it was, do you expect that different types of tropical grasses will have different emission factors?
Yeah, what we think is that the different pastures can affect different to the same manure patch. Normally, both pastures work different. Mulatto normally it has a higher nitrous oxide emission than Tule. So we expect that the manure patch and on the Tule pasture should be lower than on the other pasture. So we expect to find an effect, but it didn't happen in dry conditions because the manure patch get dry really fast. So like the effect of the BNI is on the roots of the plants, we think that the nutrients of the manure never reach the roots. So is for that never we see an effect, we think. So in what condition we could see a tendency of reduction of nitrous oxide emission from Tule. So we think that more studies have been done, but maybe could be a reduction on Tule pastures from the urine and manure patch.
Great, thank you, Banira. So let's take one more question, and this is a question for Florencia. It's from Mariana, who is at the Queen's University in Belfast. And her question is, what was the methane inhibitor that was used, and how did you test that the methane was actually inhibited? This is in reference to the treatments in the factorial design.
Thank you a lot for that question. The inhibitor was 3-NOP. That's an inorganic substance that is that is under study. It's nitrous oxide and 3-NOP, sorry. The mechanisms in which we're targeting that evaluation is that the first period is the control period with no inhibition, and the second period was the one that had the inhibitor, in which we are going to evaluate methane production and compare to the first one that was the control period. So the 8 cows were separated in 2 different diets, and within the whole period of the experiments, they had that diet, but they were divided in the group of, in the first period of time was the control, and the second one was the inhibiting period. So each cow, it's its own control in terms of methane inhibition. That's the design.
Okay, thank you, Florencia. So we have a couple more questions that we didn't get to, but as we did last time, I will ask the presenters for answers to those questions and send them out to those who've answered them, as well as share the answers with everyone else after the webinar. So let's move on, and now we're going to talk a little bit about scientific writing and publishing. And as I mentioned, if you have tips that you found really help you in your scientific writing or experiences with publishing that you want to share, please type those into the question box as we go through the presentation, and we'll discuss at the end. And I'd also like to give thanks to my colleague Todd Rosenstock for some of the ideas from this presentation. He gave this presentation during the CliffGrads workshop a couple years ago, and it was very, I thought, very stimulating and helpful. So I wanted to share some of it with you. So writing scientific papers when you're new at it. Next slide, Juliana. So first of all, my first tip is that perhaps the most important thing to keep in mind is that writing is personal. And these are some tips that I have found helpful and that, as I mentioned, my colleague Todd has found helpful, but what works for you may be something different. So over time, you will develop your own strategies. And, don't be upset or don't expect if your first article or even your second article or, your 100th article is rejected from your first choice journal. That's to be expected. Don't expect to get into science on your first try. Give yourself time to figure out approaches that work for you. Writing takes practice. And, it may be your third or fourth journal article in, you say, wow, I have been doing it, I've been really struggling this whole time, and you hit on something that really works for you. So give yourself time to write, and also, go easy on yourself. And lastly, ask your colleagues and advisors for feedback, both on your writing and also ask them what approaches and strategies work for them and how they approach scientific writing. Next slide. So here's when I'm going to tell you not to do what I do. So when I sit down to write a paper, I tend to sit there with a blank page in front of me for, a day even, because I'm trying to make it perfect. I won't write anything down until I think I've really hit on the essence of what I want to say, and I obsess over every sentence. Don't do that. It's much easier, and I find I have a much easier time if I just force myself to get my thoughts down on paper, whatever they are, in whatever messy form they are, and then rewrite. So writing is rewriting. Just get your thoughts down and then reformulate them and then make it perfect. Don't try to make your first draft a perfect first draft. Next slide. So when you're just getting started, I think a really helpful strategy is to find an article that you think is good, from your target journal that you're aiming for, or if not from your target journal, within your scientific field, and mimic the approach that article used. So what I'm showing you here is an article that I went back to again and again when I was writing one of my first articles from my PhD dissertation. It was a similar topic, and I really thought the way that it was laid out and presented was really clear and really useful. And so I essentially sort of copied the structure of that, or not copied, but mimicked the structure of that article. Every journal is going to have different guidelines. So when you do that, you want to make sure that you're following the guidelines used for the journal that you're aiming for. But it's perfectly fine if you're not reinventing, or you shouldn't reinvent the wheel with your first journal article. Mimic a format that's already out there. It's going to be much easier for you, and I think you're going to have a much simpler time in getting your first article out. Next slide. So how to get started. A caveat is that everyone has a different approach. This is the approach I like to use. So first of all, create an outline. It will change over time, obviously, but start with an outline of, in general, what you want to say. And make sure when you sit down that you have your figures prepared and you have your results more or less done and analyzed, and you know what you want to say. Don't start with the introduction. I know some people like to start with the introduction. I find it very difficult, and the truth is you're going to end up rewriting it anyway. So start with your methods or results. I like to start with the results. I think, I know what my results are. It's for me the easiest section to start with. Then work on your methods. Make sure that your methods reflect the results that you presented in the results section, and that you have included the methods for all of the relevant results that you've included in the results section. Once those are done, then write the introduction, the discussions, and conclusions. Some people like to start with the discussion and then go back and write the introduction, or vice versa, but it's helpful to write those two, together, or one right after the other, because they should in some ways mimic each other. But I find, and I think most people find, that the introduction and discussion are the most difficult sections to approach. So I'm going to give you a couple strategies for approaching the introduction and discussion. So your introduction should start with a big problem statement and then get into your specific research objectives. Now, how big of a problem statement do you start with? That kind of depends on the journal. If your journal is a more, narrowly focused journal in your field, then you want to be a little bit more narrow. But if, you're publishing in, say, Nature or Science, aiming for the big guns, or a journal that's more broad, then you want to start with a broader problem statement because the readers of that journal might not be as familiar with your field. So I'll give you an example. Next slide. So this is from an article that I published in Climate Policy. And, I'm not expecting you to read all this text, but I just want to sort of show you how the introduction is outlined. So the first paragraph starts with a big problem statement. The 2015 Paris Agreement on Climate Change for the first time commits nearly every country on the globe to lowering greenhouse gas emissions. And then it goes into, agricultural greenhouse gas emissions are highly relevant for this agreement. So, a very big problem statement. The next paragraph narrows that problem statement a little bit. Despite its critical role in future emissions, action to reduce emissions from the agricultural sector has lagged behind. And here are some potential reasons for that. The next paragraph does a very brief review, and how long this review is will vary according to the journal. Climate Policy tends to be a more pithy, short format. Since the adoption of the UNFCCC, many studies have proposed emissions allowances, et cetera, et cetera. So we're reviewing what's already out there. The next paragraph gets into the specific research objectives of this article. So we're going from, big to more narrow. Here's what we're specifically looking at. This article proposes a range of country-level targets, et cetera, et cetera. Then, explains a little bit about what we did and a brief summary of how we did it. Then the very last paragraph says why this research is novel in the context of those similar studies. So although other recent studies have assessed nationally determined contributions against targets, this is the first study to do so in this way, et cetera, et cetera. So this is sort of our inverted triangle. Big problem statement, narrow the problem statement. Here's what other studies have done, our specific objective, and then why this is novel in the context of similar studies. So this is, more or less a formula that you can follow when you're writing your introduction. And I find, for your very first journal article, it's really helpful to have a formula and just follow that formula. So next slide. Then the discussion. For me, this is the most difficult section. The introduction may be difficult, but the discussion to me is even harder. And it'll take you some practice and some time to figure out how to do a discussion well. In general, I feel that there are 4 things that you should do in a discussion. Summarize your major findings. So what are the answers to the research questions that you laid out in the introduction? Explain what the findings mean and why they are important. Then, how do the findings agree or disagree with other studies? And lastly, any unusual findings that you have or limitations to the study, you should acknowledge and explain why you might have found those findings. So let's do another example. So this is from a completely different article that I wrote actually with some colleagues at CCAFS that some of you might know. And I should say that, in this discussion, I didn't necessarily hit on all of those points. You might not necessarily include every single one of those points in your discussion, but in general, you should, and it will vary according to the journal and how much other literature is out there. But it starts with, a really brief summary of the findings. Our analysis illustrates, et cetera, et cetera, et cetera, about a paragraph. Then it gets into the broad implications of those findings. So, these findings have important implications. Other approaches that have been used, could mean this in light of our findings. The next paragraph gets into more specific summary of the findings, and the next 2 paragraphs get into more specific summary of the findings and more specific implications. So these findings have important implications. because greenhouse gas calculators don't produce absolute or relatively accurate results, application of these calculators cannot be performed with reliable results in tropical systems, et cetera, et cetera. So we're really getting into the details of the results. And then lastly, the limitations of the study. So a limitation of the study was et cetera, et cetera, et cetera. So you'll notice that it's a little less formulaic than the introduction. And, you know, that's because your discussion, it's really an art. And so, you know, you may want to use just the simple inverted triangle where you go from, you know, similar to the introduction, broad summary, implications, you know, limitations. But you may have, you know, different topics in your study that you approach, or different groups of results. So you may even have subsections in your discussion where, you know, you summarize and then talk about implications and limitations. And then different finding. You summarize, talk about implications and limitations. So it will really vary according to what your findings are, and also the journal that you're aiming for may have specific ways that they like to see the discussion written. So make sure you read the journal guidelines. Next slide. So just a couple points about the actual publishing process. First of all, dealing with revisions. I think most people know this, but I have had some colleagues who don't necessarily know this, so I think it's important to talk about. When the editor provides comments or revisions and you resubmit your journal article, which you always will, I don't think it's incredibly rare that an article is accepted without any revisions. Make sure you provide an itemized response to each comment that the editor provides you from the reviewers. It's going to vastly increase your chances of having your revision accepted without any further revisions if they can see that you've taken their comments seriously and addressed them. And in that response, clearly describe which parts of the manuscript you revised. I like to include the line numbers within the manuscript to direct the reviewers to the revisions. So, you know, my response might say, you know, thank you for this important comment. We have revised according to, you know, the suggestion in line, you know, 128 or whatever the line is. Finally, and this is something that, you know, I struggle with. When I see the comments, I tend to think that I have to change all of them. If you disagree with the reviewer's interpretation, or you think they've incorrectly interpreted or read your manuscript, you don't have to make all of the changes. You can say why you disagree. You know, obviously you want to be polite and diplomatic, and provide evidence for why you disagree with that comment or that revision and why you don't want to change it. Next slide. Lastly, rejection is completely normal. You will have so many papers that are rejected. Everyone will. Don't take it personally. You know, sometimes reviewers, those of you who publish, know that they can be pretty tough. So, you know, if you read the revisions and you find that you're having an emotional reaction to them, that's fine. Put it away for a day or two and then come back to it when you're feeling more settled about it. And then you'll be in a better frame of mind to respond, to revise, or, you know, resubmit to another journal. If it was just rejected outright, which is also very common, find another journal and resubmit it there. It might just be not a great fit for that particular journal, but there will be a journal that will publish your work. Okay, so next slide, let's take a couple minutes. And if any of you have tips for writing or publishing or questions that you have, please type them into the question box. You can also use the chat box if you would prefer. And we can have a little bit of discussion. So any questions or comments or you know, tips that you have, that includes the panelists as well. I know that some of you have published your work, so if you have things that you've found that work really well, please feel free to share them. So any comments or tips? If nobody has any, that is fine too. We can move on. Yuri says, great advice. Thank you. Maria says she prefers to start with the methodology. Thank you. Yeah, I've seen quite a few people say that is a strategy that works for them, to start with the methods. Probably because it's the most straightforward. You know exactly what you did, and you can start writing that up. So good suggestion. Let's see. Yuri says, I like to use sharing platforms. Yuri, could you type in, what do you mean by sharing platforms? Do you mean like Google Docs or that sort of thing? Can you type in maybe another comment about what you mean by sharing platforms? Like Google Drive, Dropbox, etc. Yeah, that's a really good point. When you're collaborating with multiple people, it's really helpful to have a platform like that. Personally, I prefer Google Drive because multiple people can be working on the document at the same time. But Dropbox is another possibility. Let's see, Florencia is saying, consider and have in mind that it's a long time process. Yes, that is true. And sometimes it takes a year or more to get a journal article out. But I'm hoping that some of these tips will help you do it a little bit more quickly. Let's see, people are asking that I share my presentation. I will do that. Thank you. And then Titus asks, any tips to make an outline? I think, if anyone else has outline tips, feel free to type them in. For me, I really structure it around my main findings. So, that your findings and your hypothesis or your research objective. So you can do it the same way that you would write the article. You could start with the methodology and then results, but sometimes it's for the outline, you might want to start with the introduction or discussion and really think about, what were your objectives and what are the main points that you want to make? What is really your argument, and structure it around that. Emilio, who is one of our Cliff Grads supervisors, says, do not rush and submit crude manuscripts. Edit them as many times as necessary until you get to a point at which your edits are minimal. Crude manuscripts generally irritate reviewers and editors. Yes, that is true. So you want to start just getting everything down on paper, but don't submit that version. Edit until, you really have nothing left to edit and you think it's really more or less perfect in your mind. And then submit. Otherwise, you're just likely to get a rejection. Yuri, on platforms, OneDrive, Megasync. He's saying that Megasync provides version control, which is helpful. Thanks, Yuri. Those are platforms that I actually had not heard of, so this is new for me as well. OneDrive and Megasync are platforms that he is recommending. A question from Sebastian. Thank you for the advice. When do you think it is time to share your draft with co-authors? I think that's really, to me, it's up to who your co-authors are. If they are, very involved co-authors who are going to have a hand in writing the manuscript, then I would share as early as possible. If they are, perhaps expecting to be less involved, then wait until you have some sort of draft. Well, but you do want to share your outline. So I would first share your outline and get feedback on your outline, because you don't want to write a whole draft and then have one of your co-authors say, I really don't think you should have organized it this way, or, I disagree with this finding. So share your outline, and then, depending on the involvement of the co-authors, either share an early draft or some sort of complete draft. And also, what I appreciate as a co-author is when the lead author sends the draft to me and says, here are the specific areas that I would like you to work on. I think you're much more likely to get active involvement when you give direction to your co-authors. Let's see, Maria is says, I prefer Google Drive. It has the option of suggestion. So it gets the team to get a better phrase or approach. So that's a vote for using Google Drive. From Bertin. Takutsing, from my experience, a complete cycle to get an article published is at least 12 months. That should be taken into account. Yes, really good point. So it's not just the time it takes you to draft, it's the actual publication time and the review time. So it takes quite a while. And Maria is also saying same, she uses Dropbox. Okay, so these are some great suggestions. Thank you so much. And we'll be sure to share these with everyone as you go about writing and publishing. And, if you have further suggestions, feel free to send them to myself or Juliana, and we will include them in the write-up of the webinar. All right, so let's move on. We only have a few minutes left. So thank you all so much for joining us for this webinar that exploring research approaches for emissions from livestock systems. And thank you especially to our 3 speakers. We have one webinar left in the series, which will focus on mitigating climate change by reducing food loss and waste. We're still determining the date and time for that webinar, so be on the lookout for that announcement for the last webinar in our series. Secondly, you can stand, next slide is fine, thanks Juliana. I have a bit of sad news, which is that tomorrow is my last day with CCAFS and my last day coordinating the Cliff Grads program. I've really enjoyed working with you all and watching the program grow over the past several years, so thank you for your active involvement. Juliana will be taking over, so in the future, please direct any inquiries to her. And I will be moving on to a position at Ceres, still based here in Burlington, but going to Boston occasionally. And if you would like my personal email, just shoot me an email at my UVM email and I'll provide that to you if you want to stay in touch. So thank you so much. I've really enjoyed working with you all. But before I go, I am really pleased to announce the 2019 Cliff Grads Workshop, which will be held October 6th to 7th in Bali, Indonesia. It will be just before the 5th Global Science Conference on Climate Smart Agriculture. And all Cliff Grads fellows are invited, and your costs for attendance will be covered by CCAFS, by the GRA, and the Government of New Zealand. So we'll be sending you an invitation shortly and a survey about what topics you would like to see at the workshop. And I hope you're all able to attend, both for the workshop and also for the Climate Smart Agriculture Conference. So thank you all so much for attending, and I hope you have a great rest of your week and weekend, and be on the lookout for that workshop invitation and the announcement of the next webinar. Thank you so much.